Hi everyone, this is lecture 3, and today we're going to be talking about DNA packaged into chromosomes. And this is found in chapter 2, the back part, page 21 to 30. And in particular, we're going to be talking about eukaryotic chromosomes today. First, let's start with some questions. Number 1, given a DNA molecule that was 6 base pairs in length, how many different sequences could be produced? Remember, you can pause these videos while you read the questions some more, but I'm going to keep going. Question number 2, what feature characterizes eukaryotic chromosomes? Question 3, nucleosomes consist of? And question number 4, proximity to heterochromatin can cause? So the problem is that DNA chromosomes are really, really long. The DNA, of course, is very thin, about 2 nanometers in diameter, but it's very long. Some chromosomes are 200 up to 200 million base pairs in length. Your full genome is 3 billion base pairs long. That's only the haploid genome. The diploid genome is twice that, over 6 billion base pairs long. How do you fit that much DNA into a cell which typically has a diameter of about 30 micrometers? If you were to lay all the DNA out in each one of your cells, it would be over 6 feet. How do you get that DNA packaged into such a small cell? The answer is severalfold, but the main one that we're going to talk about today is how does the DNA get wrapped into a structure called chromatin. To illustrate this problem, here is a picture of a chromosome that's been stripped of all its DNA. You'll see the scaffolding proteins that are left behind. You'll notice that this is a chromosome that's gone through replication. And so here is the so-called centromere. Here are the sister chromatids. And all the spaghetti stuff around the outside is the DNA. Here's a zoom in on this inset box up here. What a cool picture. So chromatin is the combination of DNA plus all of the associated proteins. How do you fit that much chromatin into a tiny little cell? It begins with something called the nucleosome. Nucleosomes are the basic building block of how we wrap up our DNA and to make it more dense. Nucleosomes are protein and DNA combined. The protein component is composed of these proteins called histones. Histones come in several types, 4 of which are shown here. And I'll start here on the left. 2 histones, H3 and histone H4, the blue and the green ones, will dimerize. That means they come together. Then 2 of those dimers come together to form a so-called tetramer. The other 2 here, H2A and histone H2B, the yellow and the red ones, they also dimerize and they also tetramerize. And then the tetramer of the H2A and B and the tetramer of H3 and 4 combine to form a full histone octamer. The histone octamer then is the platform from which DNA is wound around. It's about 1.5 turns of DNA. So the combination of the DNA, which is about 147 base pairs, and the histone octamer is the nucleosome. Nucleosomes can be seen in this picture at the bottom. This is an electron micrograph showing a strand of DNA with the nucleosomes aligned along at evenly spaced intervals. Here's a schematic of what that might look like. Here's the DNA, the naked DNA, and it becomes wrapped around these histone octamers, which are shown as little blue bubbles here. Again, it's about 1.5 times. And in between is another histone called histone H1. This is the first order of chromatin, first order of packing. We call this the 10 nanometer fiber because the diameter of the histone octamer is about 10 nanometers. And so this beads on a string, you'll see this model here is called the beads on a string because the nucleosomes look like beads arrayed along a string. So already we've achieved a 5-fold increase in the density of DNA just by wrapping it around a nucleosome. Going from a DNA that's 2 nanometers to one that's 10 nanometers, we've achieved a 5-fold increase in the density and a shortening of the overall length of the DNA. Of course, it doesn't stop there. The next order is called the 30 nanometer chromatin fiber, and this relies on these histone H1 proteins that you saw in the previous slide. The histone H1s coordinate these individual nucleosomes together into a so-called solenoid, or a 30 nanometer fiber structure shown here. Here is an electron micrograph down at the bottom showing this 30 nanometer fiber. So with the aid of the histone H1s, the fibers go from 10 nanometers to 30 nanometers, a threefold increase in the density of the DNA. Further looping occurs, as shown here, and here's the full schematic. Further looping occurs, these loop domains forms these 300 nanometer fibers, which then leads to the full fully condensed chromosome that you would see in a mitotic dividing cell. So nucleosomes are structural, but they also play a role in regulation, in particular with DNA expression. So how does that work? Well, histones, as shown here with these little different colored balls, have so-called histone tails, which are these long stringy things that are coming out of them. These are actually unstructured regions of the histone protein, and they're actually at the beginning of the histone protein, at the amino terminus. These histone tails have lots of amino acids on them that are basic or positively charged, for example lysine, for example arginine, that are modified by other chemical groups. So there are certain enzymes which can add modifications to these histone tails, and there's other enzymes which are going to remove those modifications. So it can be quite dynamic. Let's look at an example. This is from chapter 18 in your textbook, so I did jump ahead a little bit here. But I just wanted to show you this to illustrate how this works. Here is chromatin on the top. The nucleosomes are shown in green and the DNA is shown in blue. Here we have an activating transcription factor binding to one of those nucleosomes. And you'll notice that there is a promoter shown here in red, which is inaccessible. If this were a gene, this gene would not be turned on. However, in this case, this activator recruits an enzyme called a histone acetylase. This enzyme adds acetyl groups, C++O-CH3, to lysines on the tails of the histones. What happens here is that you take a positively charged lysine amino acid, which normally attracts the negatively charged DNA, and you make it neutral by adding the acetyl group. Now the DNA does not attach quite as strongly and becomes looser, and the chromatin begins to break apart a little bit, becomes less dense, as shown in this picture. Now this previously inaccessible promoter is accessible to other transcription factors, and the DNA can be expressed. So acetylation correlates with open chromatin and more expression. Here is an interface cell showing a cross-section through it. This is an electron micrograph showing the nucleus and the cytoplasm. Notice the nucleus has two different shades of gray. The part that's labeled E is the lighter coloring, and that's so-called euchromatin. It's chromatin that's not as dense, it's more open. Possibly it's acetylated. The chromatin that is denser, shown here with an H, is heterochromatin. That's chromatin that's maybe not acetylated, that's more packed closely together. Also, you have structures called the nucleolus, which is a very dense region of the nucleus. Sorry, NU here means nucleus. So the euchromatin is more open, the heterochromatin is more closed. You might envision this looking like this picture here, where euchromatin is more open, maybe the 10 nanometer fiber, and the heterochromatin is the looped domains, the 30 nanometer fiber. You can further subdivide heterochromatin into constitutive, meaning that it's always found constitutive, or facultative, meaning the heterochromatin is sometimes heterochromatin, but sometimes euchromatin. It's dynamic. Like I was saying, there's enzymes that can add modifications, and then enzymes that can take away those modifications. That can change during development, can happen during the stages of the cell cycle, and through differentiation and processes like that. These are some questions that we'll talk about in class as discussion questions. For example, what type of gene or other DNA sequence will be found in constitutive chromatin, heterochromatin? What types of genes might be found in facultative heterochromatin? What about euchromatin? So, like I said, we'll talk about those in class. I'll see you then. Bye-bye.